afternoon and welcome. I have the privilege of presenting to you John Ioannidis. Dr. Ioannidis holds the C.F. Renborg Chair in Disease Prevention at Stanford University and is Professor of Medicine, Professor of Health Research and Policy, and Director of the Stanford Prevention Research Center. He's also Professor of Statistics by courtesy. He is one of two directors of the Meta Research Innovation Center at Stanford, a member of the Stanford Cancer Institute and the Stanford Cardiovascular Institute, an affiliate in the Stanford Center on Longevity and the Woods Institute for the Environment. He was named one of the most influential scientists alive by The Atlantic, which also awarded him the Brave Thinker Scientist honor in 2010. Professor Ioannidis is one of the top-cited scientists worldwide and continues to create discussions about excellence in research. His thought-provoking article from 2005, Why Most Published Research Findings Are False, has been the most accessed article in the history of the Public Library of Science, with over 1 million hits. His presentation today on bias and reproducible results reflects MSU's commitment to research excellence and examines fundamental aspects of the scientific method as it is applied in the complex modern setting. Sponsored today by the Office of the Vice President for Research and the Center for Ethics and Humanities in the Life Sciences at MSU, please give a warm Spartan welcome to Dr. John Ioannidis.
Good afternoon, and thank you for the very kind invitation. It's a great pleasure to be at MSU today. I've already discussed with lots of colleagues a lot of very interesting ideas, and my mind is still buzzing with some of them. So I'd like that to be an opportunity to hear also ideas from the audience, and hopefully it's not just going to be a boring lecture. So about 10 years ago, I published a paper on why most published research findings are false. And a few months ago, trying to repent, I published another one on how to make more published research true. Both of these themes are kind of contentious and obviously hotly debated. And I will try to share some thoughts, both on the bias side, but also on the reproducibility and how we could make things better. So if we take a bird's eye view of science, we have 15 million different scientists publishing papers in major scientific journals over 15 years in Scopus, 25 million papers in that same period in that database, about 114 million scholarly documents across all science in the English language and all times, and each paper having anywhere from a few to many millions of results. So we're talking about an embarrassment of riches, a success story, a clear success story, but also an inherent burden of multiplicity of analysis and results that sometimes may be overwhelming us. How do we discern which results are worthwhile believing, pursuing, translating? Should we base our criterion on significance? Should we base it on innovation? Or should we base it on reproducibility and a rigorous reproducible approach to research? So I will try to go through these three possibilities, and significance is an obvious one. But the problem is that almost all scientific papers claim that they have found statistically or conceptually something that is significant. And obviously all the grant proposals that I have written, I say that I will find something significant, actually highly significant. Looking at Medline of close to 1.5 million abstracts with p-values, 96% of those p-values are formally, nominally, statistically significant. So everything is significant. And if you look at the trajectory, there's a bit of a decrease of the proportion of significant results in abstracts, and same thing for full text articles. We looked across about 15 million papers with David Chevalier pretty recently, and 100,000 full-text articles. But still, the vast majority of papers have significant results. So if significance means discovery, then discovery is very boring. It's all significant, it's all meaningless almost. There's no discriminating ability in just having statistically significant results. How about innovation? Innovation may be even more difficult to tackle. There's hardly any reliable method to predict innovation ahead of time. The vast majority of honest, hardworking, excellent scientists working day and night may never produce anything that is highly innovative despite all the hard work that goes into it. And innovation takes many years, often decades, to materialize and to translate itself. How about the creme de la creme of science, or for example, the most highly cited science? So we asked the most cited authors in biomedicine to tell us what they think about their most cited papers from 2005 to 2008. And we asked them how they would score them in different dimensions. So the highest scores were not for innovation, for disruption or surprise. The highest scores were for continuous progress, incremental progress, greater synthesis, broader interest, kind of expanding in an incremental, evolutionary way our landscape and knowledge rather than completely disrupting it. And if one were to analyze the correlation between features that seem to be evolutionary versus features that are revolutionary, there's no negative correlation. So that plot here, there's red links with thickness proportional to the positive correlation. There's no blue links, which would be negative correlation. So disruptive innovativeness and continuous progress have zero correlation. They have no negative correlation. And most of these pairs have some modest or even strong correlation. So these different pieces are coming together rather than opposing one another. Then we have a glacial pace of clinical translation. On average, it takes about 30 years from the first discovery until we have a highly cited clinical trial or clinical study that suggests that something would work for treating a disease or for public health. And 40% of the time, even when we get to that point, things get refuted at a subsequent stage. Here's another empirical evaluation looking at Science, Nature, Cell, Journal of Experimental Medicine, Journal of Clinical Investigation, and Journal of Biological Chemistry, and asking the question, what happened to papers published a long time ago, 1979 to 1983, in these journals, making a promise that what they found would translate to something that would change the treatment or prevention or public health implications for a disease. We found 101 such promises, and we found that only a minority of them actually went to the point of having a randomized trial downstream. Only about 25%, and even less than 20% had a quote-unquote positive randomized trial. And out of those that had a positive randomized trial, only a minority, only 5 of them, actually got licensed at some point. So the success rate is not very high, even in the most prominent journals and the most prominent promises that are being made. In fact, out of the 5 things that got licensed, only 1 of them had a major impact on that disease. The other 4 were kind of second-line treatments that are okay to have, but not necessarily big progress. What is the typical recipe of a researcher nowadays? Obviously, there's big differences across different scientific fields, but for most fields, there's a lot of competition. It means that researchers are isolated, so we have solo siloed investigators. We have small sample size studies that they can perform on their own. We have cherry picking of 1 or a few hypotheses that they can deal with. Most of the research is done post hoc. A p-value of 0.05 or slightly less is enough. No registration, no replication, and no data sharing. And what we expect to see with small studies is that we will get lots of false negatives, but also lots of false positives. So this is an empirical analysis of fMRI studies looking at 1,778 data sets. And this is the sample size on the horizontal axis and the number of foci that have been discovered in these studies or claimed to be discovered on the vertical axis. You see that the only studies that claim to have found a lot of foci are the smallest ones. And actually, when we increase the sample size, instead of discovering more, we discover less. And this is not just MRI. There's many fields where bigger studies find fewer signals, validate fewer signals compared to what we thought we had found with smaller studies. Empirically, we have looked across different domains in neuroscience. About 2 years ago, we published that paper along with colleagues from the UK and other centers in the country. And we observed that the power of studies done in neuroscience tends to be very low. In some fields, it's as low as 8 to 10%. Usually, it's 20% or less. And one anticipates that in that environment, most of the discoveries being made would be false positives and not replicated. There are some other fields where we do have replication efforts that are done routinely and at low cost. One such is human genome epidemiology, finding genes for associations with diseases. And in that case, for a long time, we were searching for one or a few genes at a time. For many years, we published tens of thousands of such papers. But then we had the benefit of having very cheap technology, of having big consortia investigators teaming together to create large efforts collaboratively. So we could test how many of our claimed discoveries in the past are actually validated based on these much larger and better conducted studies. And the replication rate across a number of such investigations on average is 1.2%. Preclinical research, the industry doesn't want to spend money for no reason, so several industry scientists have put together efforts to reproduce research, and the reproducibility rate has been 10 to 25%. In the case of Amgen, only 6 of 53 landmark studies on oncology drug target projects could be validated and reproduced. And the conclusion of Glenn Begley a couple of years ago in Nature is that the failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong. The list is very long, so I can name a very large number of empirical studies that raise reproducibility concerns in microarray data, in stroke animal studies, general biology, oncology, genome sequencing, mouse in vivo studies, mouse VPA models, genomic cell line analysis, commercial antibody studies, commercial ELISA studies, and also the way that journals handle the literature to get it published. And there's concerns that have been raised in neuroscience, in pharmacology and genomics, bioinformatics, stem cell biology, oncology in vitro testing, chemistry-led discovery, computational biology, pathology, biomarkers, organizational psychology, and any type of observational research. Going a little bit off from biomedicine, some fields have gone through a crisis in the last few years of soul searching. A couple of years ago, I was asked to write a paper for Perspectives on Psychological Science, summarizing about a dozen of empirical evaluations of research practices and what happens in psychological science. And this is an estimate of the rates of different types of discovery and replication patterns. So the optimal would be to have correct discovery and correct replication, and this is apparently happening less than 1% of the time. Wrong discovery, but then we correct it, that self-correcting paradigm, less than 1%. False non-replication, we get it right, but then we get it wrong when we try to replicate it, and seemingly doesn't replicate, but it's the replication study's fault, less than 1%. Perpetuated fallacy, the same errors are repeated in the replication study, that's 2%. And then unconfirmed genuine discovery, 43%, no one bothers to replicate, and unchallenged fallacy, we got it wrong, but nobody bothered to replicate, that's 53%. Obviously, there's a lot of uncertainty in these estimates, but last week, there was a preliminary announcement in the news at Nature, where I got this table from. The paper has not been published yet. It has been submitted. But an effort to reproduce 100 key psychology findings found that only 39 of them could be replicated. 61 could not be replicated. But then there was also the question of what exactly it means to replicate. And in some cases, maybe the results would not replicate, but maybe they were not too dissimilar compared to the original claim. And obviously, we have to wait for the final paper that's peer-reviewed before I can tell you more about it, or anyone could tell more about it. This is another field. This is animal studies in neurological diseases. This is a field where we have tons of animal studies, many of those promising that they have found effective interventions that work in animals, but very few, if any, eventually work in humans. One might say that maybe animals are not the same as humans and we cannot translate. I don't believe that. I think that for most diseases they're extremely useful as experimental models. However, when you look across more than 4,000 data sets and about 160 meta-analyses that these data sets are included in for animal research for neurological diseases, and you start counting how many of those actually didn't find anything, how many of those had very limited sample size, how many of those had just too many significant results to be true, and how many of those had evidence for bias in smaller studies giving different results from larger studies. There's very few that eventually survive out of that scrutiny. And maybe that's an explanation on why, even though we have so many promising results, eventually so few of them translate. Some people think that I'm an expert on bias, but I have to say that I know very little about bias. And a few years ago, when I tried to do that project along with David Chevalerias, we mapped 235 different biases in 17 million PubMed papers that we text mined. And most of these biases I had no clue about, or I was not even sure what do they mean. I tried to think when I designed a piece of research of what might the key biases be here that might be operating. But I have to admit that it's very difficult to anticipate all the possible biases that may interfere in the process. And I think that most of us are not so much trained to think about the bias component. We're mostly trained about the subject matter and trying to get a significant result rather than what necessarily could have gone wrong. And it's just that there's a vast array of possibilities that could go wrong. This is not a microarray experiment, it's a micro-bias analysis. I'm just showing you a heat map of the occurrence of bias terms in the biomedical literature. There's also an error here. If you might notice, this is 1882. Well, it's a scientific work. There has to be some error somewhere. Actually, the range is from 1960 until pretty recently. If you see dark color, it means that these biases are not discussed. If you see white color, it means that they're hot in the literature. So lots of papers deal with them. They're discussing them. They mention them at least. So confounding and selection bias and publication bias have been very popular in the last couple of decades or more. The question is, what does that mean? If you look at the papers, they do mention confounding. So lots of epidemiological research, almost all observational studies, they would mention confounding is an issue. But our study doesn't have that problem because we took care of it. Or if you read meta-analysis, they will say publication bias is an issue for meta-analysis, but we don't have a problem in our paper. Or selection bias is a problem in surveys, but we took the utmost care to get rid of selection bias. So we think of these biases, but it doesn't mean necessarily that we admit them, that they may be influential. Some countries may be even worse. So along with Dan Fanelli, who recently joined us at Metrics at Stanford, a couple of years ago, we looked at US studies versus studies done with corresponding authors in other countries. And we compared genetics, where for whatever biases they may have, the measurements are very accurate, and you either have a genotype or not, versus psychology, psychiatry, where the measurements are behavioral, and therefore there's more flexibility and some leeway on how to define them and capture them. We saw that US studies tended to give more exaggerated results whenever we had behavioral outcomes and behavioral measurements. And maybe one possibility is that our study is false and it will not replicate. But another possibility is that this is genuine, and maybe there has been more pressure on US scientists to deliver more extreme results, and this is easier to do when the outcome or the measurement is more subjective and more error-prone and more flexible in the way that it will be analyzed. Obviously, these are data that were included in meta-analysis, and they belong to the past. On average, maybe they are 5, 10, 15 years old. I think currently there's pressure on scientists to deliver significant results and extreme results all over the world. I think we've seen patterns in some cases in research done in China and research done in Europe. So I think that this is an evolving landscape. Another parameter that has changed is the advent of big data. So big data is a big promise. I'm very sympathetic, and I run lots of analysis that involve big data, but at the same time, they can lead to big noise and big error. Along with Moin Khoury, we tried to discuss these issues in a short paper in Science a few months ago. I don't want to dismiss that potential, but you can imagine that by escalating the space of analysis by several fold on a log scale, it really creates far more opportunities for false positives and for erratic results that may start populating the literature unless we have a clear plan on what to do with them, how to validate them, how we take them to the next step. In some cases, we don't see many of these results. A few months ago, I discussed the phenomenon of stealth research in JAMA, where lots of very innovative and very smart and brilliant entrepreneurs may try to develop a technology, they may patent it, and then it's out in the wild world, but actually there's no scientific publications to document how exactly this has happened. And just going to the patent and trying to understand from the patent what has happened, it's not going to work. We need some better transparency in order to be able to judge the real potential of this research. I'll spend the second half of the presentation on potential solutions. And I'm very happy to see that there are more and more solutions being proposed. At the same time, many of these solutions sound really great, but they're very speculative. We have solutions that have worked in specific fields, and maybe we could take them and apply them in other fields as well. This is an open question. And those that have already worked have a better chance of working again. But others are just theoretical, and sometimes we have to be careful because we need evidence on solutions about the problems. I mean, just saying that I will change something does not mean that I will make it better. So not harming the scientific enterprise should be a priority, just as it would be a priority not harming patients when it comes to medicine and medical research. The solutions have to face the whole spectrum of problems that start from thinking about a study, designing it, conducting it, analyzing it, and reporting it. And I've told you a little bit about what the problems are. There are multiple, and I just mentioned a few. Then there's lots of potential actions that could remedy some of these problems, but we have to think what would be the collateral impact also on other aspects of the research flow. So in the PLOS Medicine paper, I tried to categorize these solutions into 12 categories, and I will discuss some of them in more detail based on my biased perception of what might work best. I'm a believer in large-scale collaboration, and I think that I have seen it be successful in enough scientific fields that I think it has proven that it's the way to move forward. Probably biomedicine is still lagging behind compared to the physical sciences, but we have seen tremendous evolution, for example, in genomics and many other omics fields. And this is one example from a meta-analysis of multiple teams joining forces to perform GWAS studies, and then a meta-analysis of them in a large consortium of smoking behavior. So before we did that, we thought that we had found 659 genes that regulate smoking behavior. And they were in the scientific literature, in reviews that were updated every year or so. And when we did that collaborative study, none of them really survived. We found 4, and now we're up to about 10 for smoking behavior. And in some other fields, we have been more successful. Is it bad that we went from 659 down to 4 or 10? Well, if this is what it is, this is what it is. I think that at least we know that these 10 are genuine, and we can take them to the next step and see whether they can give us any other insights on their biology or mechanisms of action, rather than following leads on 659 genes that were not really leading us anywhere. The next question is replication. I'm a big believer in replication, but then the question is who is going to do it and how is it going to be done and when? One option is to have the same investigator, so you have a consortium like the one that I showed you before. So replication is part of the primary research. It's kind of indistinguishable from primary research, and that might work. In some cases, one might argue that you need some contrarian to try to replicate. So CERN has two teams that are working collaboratively, but they're also checking each other. And then you have different possibilities. You can think of investigators of the same school of thought, but then you may be running into some sort of inbreeding and people getting the same results if they have been trained and educated and they have built their career based on some theory or some scientific findings. You may have truly contrarian replication from investigators of competing theories of hypotheses. People have actually, their stake is to refute the new theory or the new finding. You can have combinations of the above, or you can open replication to the wide and wild public and think that everybody in the community could be a scientist. And depending on the question, in some cases, this might be possible. So for questions that don't require a lot of technical expertise, but you just need to devote your computer to run some code and see what happens. Maybe this could be done with crowdsourcing in very large scale. But I think we need a little bit more evidence on how exactly these different options work and how they operate in real life for different applications. Registration is another possibility. So registering ideas, protocols, analysis ahead of doing them or evaluating them. It's something that has worked in some fields. For example, clinical trials have been transformed in terms of their transparency after the launch of ClinicalTrials.gov and several other registries for clinical trial, and the request by all major journals and also by ClinicalTrials.gov that all clinical trials should be registered. So major clinical journals are not going to publish a randomized trial unless it is registered ahead of time before it's done. Still, most trials are not registered. So recently we saw that about 70% of non-drug trials are not registered, and they account probably for about 50% of all trials. And even for drug trials, because many of those don't have strict regulatory requirements and they're not into some regulatory pipeline, a big share of them are still not registered. How about other types of designs? How about non-randomized research, observational research? There has been a lot of debate. And people have claimed that maybe it's not possible to register such studies. In many cases, these studies don't really exist. If one has a database and goes to that database and start running analysis, lots of chi-square or Fisher exact tests, is that a study? If I force investigators to say that that was a study, I think that I'm forcing them to lie. It would be very misleading. It will sound as if they had that preconceived and very carefully thought out, while it was entirely exploratory. So what I have argued is that I don't care about these studies. I care about studies that could exist but actually don't exist, and I would like to know their potential for existing, which means that I would like to know about what data sets are available, what opportunities are there for analysis to be done, so that when I see one analysis being published, I have some sense that maybe there's 10 other teams or 100 other teams or 5 million other teams who could do the same analysis. To give you an example, recently we looked at the literature on associations between cancer and diabetes, different types of cancer and diabetes. For most types of cancer, we could find anywhere from 5 to 70 studies. How many data sets are out there that could evaluate whether there's an association between cancer and diabetes? I would argue that any hospital database anywhere in the country or anywhere in the world could look at that. Probably there's tens, hundreds of thousands of data sets that could potentially inform that question. Not all of them very well data collected, not all of them high quality, But in theory, the denominator of potential analysis is really huge. And therefore, one may think of different levels of registration. You can think of level 0, where I admit that this is something that is not registered. I just had that idea, I had that data set, and I just wanted to test it. And here's what I get. I think it's perfectly fine to do this type of exploratory research, but then it has to be presented as such in a transparent mode, and it also means that someone needs to replicate it. And now we do have a plan that we specifically want to test that idea that John had, and almost likely, 100% likely to be false. But at least we have a clear pathway to test it. In some cases, it will not be false. In some cases, maybe the best discoveries that we can make out of just exploration. Level 1 would be registration of data sets. So we know the potential of a data set to run different analyses, what kind of variables are included, what could have been analyzed or could be analyzed in the future. Level 2 is registration of the protocol. Level 3 is registration of the analysis plan, which is different than the protocol. It's far more specified, and in many studies, even asking very seasoned statisticians, they admit that they specify the analysis plan at the end of the day, when the study has been completed. And the rationale for this is that, well, many things could happen while the study is being conducted. There can be debate about that. I'm trying to push more in the direction of having an analysis pre-specified. At a minimum, I think we should know whether an analysis is pre-specified or was decided at a late stage. And if it was decided at a late stage, when exactly was it decided? Level 4 would be registration of the analysis plan and raw data. And then there's many layers of what we mean by raw data. So there's super processed and trimmed and cleaned raw data. There's processed raw data. There's unprocessed entries. There's data forms. There's sometimes even other layers of raw data. And sometimes these layers may not necessarily agree with each other. So we need to decide what exactly is convenient, efficient, and who should do what if we want to share raw data. And level 5 would be open live streaming. I open my computer. You can see everything that I plan to do. I tell you that today I'm going to run these experiments. So in the case of the claim that NASA had discovered arsenic bacteria that could use arsenic instead of phosphorus for their DNA backbone, at least one researcher had an open live streaming where she would post all the experiments she was planning to do. People could comment on that and then show all the results. This is what I got today. This is what I'm planning to do tomorrow. And eventually the claim could not be reproduced. How much of that is happening already? People are sensitized, and journals and funders are sensitized. A couple of years ago, we looked at the 50 highest impact factor journals that publish primary research across all sciences. There's a heavy infiltration of biomedicine here, but also physics and chemistry and biotechnology and other sciences are represented as well. And we tried to see how many of those had policies in place for sharing microarray data or sharing other types of raw data or requesting materials from the investigators or asking for protocols or even making that a prerequisite for publication, that unless some or all of that is available in public view, then we cannot publish the paper. And you see lots of green color in that slide. It means that that's good news. The journals were sensitized and they did have policies in place. But the last column, which is very tiny font, you have to trust me that there's lots of zeros there. And zeros means how many out of 10 papers that we evaluated from each of these journals actually implemented these policies. So even though the policies were in place, in reality, this did not happen. Along with colleagues from NIH, we have repeated that exercise with data from journals publishing in 2014. It looks a little better, but not much better. And also, we have performed an analysis of a random sample from PubMed covering the last 15 years. And we found that in the big scheme of things, not just the top journals, but picking any journal that would find its way into PubMed, out of 500 papers, only one of them had raw data available. And only one of them had a protocol. Actually, it was a protocol publication. and very few of them had active efforts for replication. So even though these ideas circulate, I think that they still have not captured the majority of the scientific literature. Sharing is easily said and more difficult to do, I have to acknowledge. And I think that even though everybody would, or almost everybody would say, that's an interesting idea, when it comes to actually sharing data and when it actually comes to sharing my own data, I have to think, when am I going to do that? Who's going to fund me to do this? Do I need to clean further the data? Will someone else understand what I did here? Do I need some extra orientation? Do I need to spend 10% of my time from now on for each paper that I do to explain to others how I did it? This means that at 10 papers, I'm out. That would be the rest of my career. So it's not easy. And this is a list of about 25 challenges on how exactly to share data. Nevertheless, we do have success stories. I think that we have plenty of success stories from the physical sciences. We have success stories in genomics and other omics fields. And we start probing the space in different types of studies and analysis. Once you have access to raw data, maybe we will have a generation of repeatability scientists. You have nothing else to do in your life. Well, you can always reanalyze an existing study. Maybe not a bad thing to do. So Keith Baggerly spent 4 years reanalyzing the data on molecular signatures from Neil Potter's team. Nobody wanted to publish his results, even though he was saying, but I cannot reproduce anything of what has been published in all these major papers in major journals, until it was found out that Anil Pati had claimed to be a Rhodes Scholar and he was not, and then an investigation started, and indeed people realized that all these papers were wrong and none of these molecular signatures were correct, and the several clinical trials that had been launched at Duke had to be stopped. So this type of forensic bioinformatics approach may be appealing, and it may have some use. This is one experience that I had in collaboration with Nature Genetics. Nature Genetics has been wonderful for many years now. They ask for microarray studies to be published only if raw data, any processing of data, protocols, algorithms, software, codes is available in public view. Otherwise, the paper is not going to be published. So we looked at 18 papers. We had several teams of microarray experts around the world trying to reproduce and reanalyze single tables or figures from each one of these 18 papers. And apparently, we could do that only for 2 of the 18 papers. In some cases, we could do it with discrepancies or from process data with discrepancies or partially with discrepancies. In the majority, it could not be reproduced at all. And the reason was that the data were not available, even though it was supposed to be there. The software was homemade and it had disappeared in the meanwhile. The methods were unclear, or everything was very clear, but the result was different. This is the highest impact factor journal in genomics and one of the highest impact factor in the world. Very high quality research. All the teams that we analyze paper, I think, are stellar scientists. I'm not saying that they're worse than anyone else. Probably they're better than anyone else, and definitely better than my own research. But still, this is the type of repeatability that we got. A few months ago, we decided to look at another field in terms of repeatability and reanalysis, randomized controlled trials. There's a lot of push to try to make data from randomized trials publicly available. There's clearly rationale for that. There's even, I would argue, an ethical reason to promote this. These people have volunteered for the common good, and I think that these are data that are valuable. People need to see, they need to be able to check what was done, they need to be able to use it for additional insights and combined analysis. So this has happened to some extent already, but not extensively. So we try to find across PubMed any opportunities where reanalysis of randomized trials had been done. We found 37 such reanalysis of different randomized trials where the raw data had been reassessed, looking at the very same question, the same outcome as the original study. 35% of the time, the conclusion was different, not in subtle ways, but in terms of whether the treatment should be used or not, or who are the patients who should be treated. And the next question is, well, maybe it was the enemies of the original investigators who wanted to put them to shame. But actually, almost all of these papers, they included authors from the original team. So why would someone contradict their own initial findings? My explanation for that is that in the current environment, a reanalysis cannot be published anywhere unless you claim that you have found something different. So, and maybe the 35% is pretty low. So you need to kill your study, and it's interesting, you know, people don't say we were wrong. They say we gain new insights, but when you read it, well, you know, based on the first paper, I should use this drug. Based on this paper, I should not use this drug. So is that new insights, or I'm just confused? So I think that we need to readdress what are the incentives behind running these reanalyses, and why these incentives might need to change. Related to incentives is also the question of who should be doing different types of studies and who should be sponsoring different studies. So there's clearly lots of areas in biomedicine, but also some other scientific fields where there are conflicts of interest, beyond just academic and scientific conflicts of interest. I want to prove that I'm correct. There's money to be made. And I think that we need more transparency in showing potential conflicts of interest. This has improved. In our analysis of the biomedical literature, we see that there is more papers reporting conflicts of interest and fewer papers that say absolutely nothing. Saying that there are no conflicts of interest is something over time. So this is good news, but still, I think there's lots of conflicts of interest that are not transparent, and this has been seen in multiple empirical investigations. Then the question is also, who should sponsor sensitive studies that have clear impact not only on finances, but also on human health, human lives, human well-being? Randomized trials, meta-analysis, cost-effectiveness analysis, guidelines. Should they be sponsored by the industry? I would argue that probably not. Maybe that money that the industry is putting into sponsoring such trials, maybe they could invest into some more translational, entrepreneurial discovery type of research. And conversely, a small portion of that money may be sufficient to sustain a federally decided agenda by unconflicted authors who have nothing to gain, at least financially, by deciding what are the questions that we need to ask and how exactly are we going to ask them for these types of designs. This is what's happening currently. It's an auto-looping system where all research being done is done as if each company is the entire medicine or the entire science on its own. To give the analogy, talking to the Spartans, it's like the ancient city-states. Each city-state was alone, and it thought that it was the entire universe. So this is 600 trials sponsored by the 15 most prolific companies. If you see a node, a blue node, it's a company, and the size of that node is proportional to the number of trials that they have sponsored. If you see a connection between two nodes, this means that there are trials that involve drugs or interventions that these connected companies have. So if you see a disconnected, a discontinuous line, this means that there's just a single trial. If you see a line that is a bit more thick, there's a couple of trials. Here's Sanofi and BMS. They had co-manufactured a drug, I think clopidogrel, and therefore there's kind of a more thick connection. But almost all of the action is not connections between these companies. Almost all of the action is these auto loops, with the thickness of the auto loop being proportional to the number of trials, where only agents, drugs, regimens, biologics of that single company are involved. And it's not necessarily my drug versus placebo, which is one option. It could be my drug 1 plus my drug 2 versus my drug 2. So if the first arm ends up being better, then this means that both drugs could be marketed. If the second arm ends up being better, it means that drug 2 could be marketed. If the two arms end up being non-inferior to each other, then again, all drugs could be marketed. So no matter what you get in terms of results, it's good news for the company. And there's very little evidence where we see head-to-head comparisons where one company is risking to lose its reputation for its product against a competitor. When it happens, surprisingly, we always get the right answer. So recently we looked at trials published in 2011. 55 out of 57 of non-inferiority trials with head-to-head comparisons sponsored by the industry demonstrated non-inferiority had favorable results for the sponsor. The success rate was 96.5%. I would argue that we don't need these trials. If we have 96, 97% success rate, we can skip that step. We can just say, oh yeah, it works. It has non-inferiority. It's good enough. So I think that the results would have been different if we had someone who was not conflicted design that agenda. And I think that this is something that needs to be revisited. Meta-analysis have the same problem. So I have been a very strong supporter of meta-analysis for many years, and I have been very excited about the prospect of joining teams and data sets and combining results across multiple studies. But you can get the same problems, and sometimes even worse. So recently, we looked at meta-analysis on antidepressants for depression. I wanted to do that project, and I convinced Shanil Ibrahim and several other fellows from my team to work on this, because I was seeing just lots of these meta-analyses all the time. And we found 185 meta-analyses over 6 years. Why do we need 185 meta-analyses just for a single indication for a single type of drug? There's different drugs there. Maybe we need 6 or 10 or 12, but not 185. About 30% of them had authors who were employees of the assessed drug manufacturer, and about 80% of them had some industry funding or other link. Only 30% had some negative statement in the concluding statement of the abstract about antidepressants. And when you split these meta-analyses according to where they came from, only 1 out of 54 meta-analyses that had industry employees had any caveat, any negative statement in the abstract about antidepressants. Actually, I don't know whether these employees are still employed by that company. But conversely, when you look at meta-analyses that had absolutely no industry involvement, about 50% of them had negative statements and caveats about antidepressants. So even though meta-analysis supposedly should be better, in that conflicted environment, it's very hard to say whether I would believe it or not. And then guidelines. Guidelines could also be affected severely, and there have been hot debates in the last several years, resurfacing again and again, on whether guidelines were conflicted and whether they can be trusted or not. So about a year ago, along with several colleagues, we tried to put together a number of suggested red flags that people should consider when they read a guideline. And unfortunately, I have to say, most guidelines currently, especially those that are very influential, they carry at least one or more red flags, so one has to be very careful about how to interpret their recommendations. Better statistics and methods. Clearly, that could make a big difference. And I think that there's no end on how we can educate better our undergraduate students, graduate students, young researchers, mid-career researchers, late-stage researchers on better statistics, better tools, better methods on how to design, analyze, and present results. There's several empirical studies that suggest that there's extensive illiteracy and innumeracy among scientists, and it's even worse in the general public. So the general public trying to understand scientific results in medicine or other fields is really at a great disadvantage. And then the question is how to do that. It's not necessarily very easy. Maybe sometimes we can force some sine qua non requirements that you need to do these 5 things. And NIH has been experimenting in the last couple of years with checklists that at a minimum you need to check off these boxes if you want to do this type of research. For example, some features could be standard: randomization and blinding of investigators in animal experiments. They're easy to do. They should be the default. However, if you look at empirical data, this is how many studies do this. So blinded assessment of outcome, randomization, and I also show you some data on statements on conflict of interest. even though there is some improvement, still in the most recent cohort of studies, the rates are about 10 to 30% for these practices to be implemented. So there's plenty of way for improvement here. Institutions could make a difference also. I think that some of that educational component could be undertaken by institutions, and it could happen at multiple levels. It could happen at the levels of students and postdoctoral fellows. It could happen at the level of investigators. It would happen at the level of institutional policies in general on how research should be conducted. Here's a list of potential proposals that have been raised. I have to say that I'm sympathetic with some of them, interested in most of them, skeptical about some others. So for example, one proposal is that an institution should have a police force, and that police force should go and check notebooks and experiments. But then the question is, who is going to be in that police force? If they really have to understand the science, you need probably an avatar of each investigator who would be, you know, his police avatar of reproducibility to even understand what that guy is doing. I mean, it would probably take retraining in a new career from scratch. So I also have to say that I'm very worried about solutions that are not driven by the scientific community itself, but are imposed as yet another administrative hurdle. That, oh, well, I had to satisfy all of these requirements, and now I have to do that. Not because I believe that my science will be better, more reliable, more reproducible, but because I have to satisfy a different entity. So I think that we need more evidence on many of these ideas. At the end of the day, maybe we just have to learn to live with small or even tiny effects. And by that I mean that if you're an epidemiologist like myself, I don't expect to find in my career so many discoveries of the range of smoking and lung cancer. You know, I may find 20 of them, and 20 of them would be wrong, most likely, because these are gone. There's 15 million scientists before me, and they had all the opportunity to find about them. So why did they wait for me? Probably it's unlikely that I will get them unless there's really some new disruptive technology, and all these 15 million scientists could not really look at them before. So effect sizes are becoming smaller and smaller. A couple of years ago, we cataloged a number of risk factors and interventions that have relative risk less than 1.05 on a relative scale. And one might say, well, do I care about that? Maybe we don't need to worry about relative risk less than 1.05. In some cases, yes. Maybe we just have research that has no translation potential, but in other cases, no. So if these things are pretty common, they could have a big population impact. For example, fruits and vegetables. I'm directing the Stanford Prevention Research Center at Stanford. My chair is on disease prevention. I advocate that it's wonderful to eat more fruits and vegetables, and I would tell you again that this is a good thing. The studies suggest that for cancer, the hazard ratio for one serving of fruits or vegetables per day is 0.998 or 0.999. So we're talking about 1 in 1,000 or 2 in 1,000 absolute reduction in cancer risk. Is that worth it? It is worth it, because at a global population level, we're talking about tens of thousands of cancers and deaths that could be averted if that 0.998 or 0.999 is correct. So I think we should compromise that there's lots of interesting things out there that are just very small, and this means that we need very carefully conducted studies, minimize noise, minimize error, to really have a chance to figure out these tiny, subtle effects. This is another piece of evidence along the same lines, looking at medical interventions across all medicine. A couple of years ago, we looked at the entire Cochrane database, close to 85,000 forest plots of meta-analysis, including 220,000 plus clinical trials worth of data. And we asked the question, how often do you see across, this is about 60% of all medicine, because Cochrane by that time had not covered everything, but it was a big chunk. How often do you see an intervention that decreases mortality 5-fold, that this has been seen in 2 trials, and with significant results, less than 0.001, and with no bias, at least florid overt bias. We could only find one such intervention, extracorporeal oxygenation for premature babies. There are some more. Paul Glasziou has tried to put together a number of interventions that have never been tested in randomized trials because they are so effective that who's going to test them? But they are still very limited. Probably we're talking about a dozen, maybe a couple dozen, out of hundreds and thousands and tens of thousands of interventions in different settings that have been tried. Conversely, we do see very big effects. We do see relative risk more than 5-fold reductions in major outcomes in small trials. We see that pretty often. About 10% of small trials have these huge odds ratios above 5. However, 99+% of the time, when we run a bigger study, that goes away. Typically, it goes completely away, it evaporates, or in some cases, it becomes a much smaller contained effect. When you come to epidemiology and you have no randomization, I think that extreme effects are even more problematic. These are clouds of results for epidemiological associations that we have generated in data from the National Household Survey, NHANES. And they pertain to associations of vitamin D, thyroxine, creatinine, and vitamin E in association with mortality risk. So why do we have clouds of results instead of a single result? Because for each one of these associations, we have 1 million ways that they can be analyzed. How do we get 1 million different ways? Well, this is very easy. There's lots of things that can affect whether people die or not sooner or later. And if you want to account or not account for each of 14 variables, you have 2 options for account or not account, another times 2 for accounting, not accounting. If you run through that, you end up with about a million choices. And you get a million different results. Very different, both in terms of statistical significance and in terms of effect size. About 40% of the time, you get that Janus effect picture, where for vitamin E and many, many other things, 70% of the results suggest that vitamin E reduces the risk of death. 30% of the results suggest that vitamin E increases the risk of death. So depending on what you like, you can pick your result and publish it. And then you can also publish a replication or multiple replications, just sampling out of these millions of analyses. Improving research reporting standards could also go a long way towards transparency. If you go to the EQUATOR site that Doug Altman and his team launched several years ago, there's at least 240 guidelines, recommendations on how to report different types of designs. They're happening after the fact, so people now have started creating also equivalent reporting guidelines for protocols rather than just the study that has been finished. But at least I think it's a step in the right direction in that we can now understand at least what has happened. One might argue, well, if something was done in a suboptimal way, would people acknowledge that? There's some debate about this. And this is one more reason why some pre-registration, especially for results and for studies that do have the potential for pre-registration, would make more sense. Then there's lots of efforts for reproducibility checks. And the Center for Open Science, led by Brian Nosek, is one of them. The reproducibility initiative that Elizabeth Irons is leading is conducting reproducibility checks in cell biology and in prostate cancer research at the moment. There's several other efforts that are trying to map the reproducibility profile in different scientific fields. And I think that these probes are very useful because they can give us some guidance of where we may have more major problems and where things may be okay. I would argue that we don't need reproducibility checks on everything because obviously that would take enormous effort. But I think that we do need these projects that would give us a sense of where different scientific fields lie, what is their performance characteristics. And I think that we also need them in cases where the research is very influential. So for example, a piece of research that is used by many other researchers to build their own research on, or research that leads to clinical experimentation, humans are exposed to risk, even worse, humans are being treated in massive scale. I think that type of research, before we take these extra steps, needs to be reproduced. The new center that I launched with Steve Goodman and several other colleagues at Stanford about a year ago the Meta Research Innovation Center at Stanford, is trying to create a connector hub for all of these initiatives. There's lots of initiatives happening around the globe. I find out about more of them almost on a daily basis. They're happening in very remote fields that probably do not communicate much with each other. And I think that we have to learn a lot. I think that astrophysics, for example, may have lots to share about how they collect data and distribute data from large telescopes that could be of use to medical sciences. Genomics could teach a lot to nutritional epidemiology. Economics has a lot of literature on model specification that probably is not very well known in biomedicine. So I think that these themes are common, they're pervasive, and probably being able to bring different scientific disciplines together and make them communicate and share information and insights would be useful. I will close with some suggestions on the reward system and changing incentives. Until now, most of the funding agencies, most of the journals, most of the reward mechanisms focus on getting significant, extravagant, extreme, fancy results. So this is what we reward, this is what we get. But I don't think that this is really what we get. We want results that would be reproducible, we want results that would be translatable, we want results that would be real. So I think we need to reappraise what has value and maybe re-engineer the reward system. One possibility is to think of the PQRST. It sounds like an electrocardiogram, so it's the electrocardiogram of research reward. P stands for productivity, and I think that we do reward productivity currently, but I would argue that maybe even productivity could be revisited. So instead of looking at number of publications, that doesn't mean anything, especially in the current environment with predatory journals. I probably get about 20 invitations to publish my paper by the end of the day. And you know, I could have had probably 7,000 papers per year easily if I had the money to pay for that. But instead of just counting papers, I would say we count some other qualitative indices and maybe focus on the high impact papers, the very few papers that do make a difference. If it's not papers about protocols, I would say we can judge whether an investigator has registered his or her protocols. If it's funding, we can see, I got that funding. Did I publish based on what I promised to do? And if I didn't publish, maybe I shouldn't be funded again, or unless I have some reason why I didn't publish anything. It could have been negative results, but you know, I spent some money to do this. Q stands for quality. And I think it's the scientific community who should decide what is essential for quality. Probably it's not going to be the same in different scientific fields. So I mentioned animal research. I think that the community does understand now that blinding of investigators to reading outcomes and randomization is important. So I think that there can be some consensus that we will judge you based on whether you do fulfill these requirements. But each field needs to decide what are really the essential, quintessential quality aspects that are inherent to better science. Because I can think of hundreds of quality lists and checks that would have no evidence to support them, and things could go really weird. R, reproducibility. So reward scientists for reproducible work. If my paper gets replicated, then I can get extra credit, or maybe I lose credit if my paper does not get replicated. S, sharing. We offer credit for sharing of data, of protocols, of algorithms, of codes. There's increasingly a number of efforts to try to capture some of that credit allocation. But it's still, I think, in early days. And I think we can do better in giving credit for sharing. And finally, T is translational impact of research, which I would argue does not apply to people who are doing blue skies, early discovery research, because who knows whether it will be translated and when and how. But for applied researchers, I think that there should be an element of, did that work eventually? And did it lead to something meaningful that could be applied in real life and practice? So you can think of these value systems as a stock exchange, where different items in the research world have value of high value or low value or no value. So you can think of currencies. The current situation is that the paper has value. It doesn't matter whether it's replicated, refuted, translated, it's all the same. More papers. Sharing probably has very little value. Contribution to peer review, meaningful contribution, very little value. Contribution to education in a meaningful way, again, no value. Grants, they carry value. So here I say that 1 R01 is 5 points. So 5 good publications, you can exchange that with an R01 or vice versa. And then these values, they can buy other wealth items like promotion. So an assistant professor is worth 3 good papers. An associate professor, 10 papers, a tenure professor, 20 papers. And obviously different universities will have more or less stringent rules about that. If you have students, each student is worth 2 points, which means that you can trade 1 student and get 2 papers, or trade 2.5 students and get an R01. It's like a slave market. And administrative power could be anywhere from 0 to 200 points. So, some sort of lobbying and networking could get you money that may be worth 40 R01s, and you just sit at your office. You need to give me that money. And, that's all you do, and that's good enough. So that pattern may be possible to change. So the change 1 scenario here, papers do have value, but they have more value if they're replicated. They have less or no value if they're refuted, or even negative. You even get a -1 for a refuted claim. And successful translation carries even more value. Sharing data has value, contributing to peer review. Education also carries value. And then you have a more disruptive change, which is change 2, where you have no ranks. Everybody is equal. There's no professors, no students. Everybody's a scientist in the community. If you get a grant, you lose points because, oh my goodness, I got a grant. What am I going to do now? I need to deliver based on the money that I got. And if you have other types of power, you also lose points. So if you follow that paradigm, then nobody will want to submit grant proposals, and nobody would want to become a leader, or almost. So I'm not saying that we need to go to extremes, but we need to think about how to fine-tune and how to exactly find what is really worthy, and then we should go for it. Finally, we need to see who else is in the same business or in the same endeavor and enterprise. Some people see science as a calling, as pure excitement. Others see that as money. Others might see that as a business or something in the middle. So it's not just scientists. There's industry involved. There's private investors, public investors, funders, not-for-profit philanthropists, journal editors, for-profit publishers, insurance companies, scientific societies, professional societies, universities, research institutions, hospitals, patients, people who are interested in science. And they have very different views on whether they want scientific work to be publishable, to be fundable, to be translatable, or to be profitable, or combinations of these possibilities. And even though I would argue that my viewpoint has always been that science is the best thing that can happen to human beings, and it's a very impressive endeavor, very difficult, and has very lofty ideals, I cannot ignore that there's lots of other stakeholders that don't think this way. And we have to take into account what their priorities are and how we can align their priorities with that lofty idea of producing the best, most reliable results that can also have an impact on fellow human beings. To conclude, I think that there's lots of ideas out there about how to improve the scientific enterprise and research practices. There's many possible interventions that have been proposed. Some of them may improve really the efficiency of what we do and the reproducibility of the literature. However, I still have to admit that few of these proposals have strong empirical support. And I think that we need more empirical support, especially when we have competing proposals on how to move forward and how to change the world. Many of these ideas may be very beneficial, but some of them may be harmful, and I think that we need to be able to sort these out. Eventually, the adoption of the best, the real optimal research practices can be key to the success rate, efficiency, credibility, and legacy of scientific fields and science at large. So I think really there's lots at stake here. That's a list of some of my collaborators that I want to thank for some of the empirical work that I showed you today. I think that nothing of what I showed you in terms of empirical data would have happened unless I had the benefit to learn from them and to see their hard work materialize. Thank you.
We have 2 microphones at the front. Anybody who would like to ask a question, just come to the front and queue up.
Oh, I don't think I need a microphone. We, Lasagna, pioneered the idea that you should have clinical trials enforced by government and set up these institutions to screen out harmful things and control the industry. And late in his career, he went from being much liked for that point of view and actually pushed it through, being a bit of a goat because he suggested that Marilyn Monroe probably wouldn't have died if she'd been prescribed thalidomide. So he thought somehow they'd gone too far. What would you whisper in his ear if you'd been, had his ear back then about, well, the risks of, for example, ignoring the fact that women might be different in their response to medications than men, and all of these huge gaps that have gone on for decades, and nobody seems to have cared or noticed. We need more room for skeptics, I would say. So I ask you again, what might you caution about this?
So clinical trials do have limitations, and you know, they will not be necessarily the final answer for everything. However, my stance is that we need randomized, well-designed randomized trials on relevant, pragmatic outcomes, sufficiently large to be clinically meaningful, to look at the right questions. And I think that these trials should be done by non-conflicted stakeholders. So I think that it should belong to the public domain, their conduct analysis and dissemination. Even more so, their basic design of their research agenda and what are the questions that we need to ask. This doesn't mean that there's not some questions about effects in specific populations, men versus women, or subsets, or specific types of patients that other types of design could inform us about that may be very difficult to wait for clinical trials to answer all of those. However, the experience that we have, for example, for subgroup differences, like effect sizes being different in men versus women, most of the claims that have been made have not been validated. And if one looks at how these claims have been made, they have been made in an environment where the analysis was underpowered, because the clinical trials available were powered for the overall analysis, not for detecting subgroup differences. So I'm a little bit worried about the eagerness to generate lots of specific effects, especially now with precision medicine being such a buzzword, on small groups of patients who would have very different benefits and harms compared to the average that one sees in a clinical trial. I think we need more of that, but we still need to map the operational characteristics of this type of research.
Over here.
So I enjoyed your presentation very much, but it left me with a couple things unsettled. So one, it's that there may be other value in 90 or 95% of those papers that get refuted. They could stimulate other researchers with some different ideas or present methods that can be employed in other fields. So there may be some value in those studies that you suggest to throw out. And the other is that I forget who made that quote, but stated that hypothesis is only as good as it can be refuted and a new hypothesis can be created. So in a way, we advance in a way that we refute hypotheses that don't work and come up with better models. So if we don't publish those papers, we're not going to have progress.
These are both very interesting comments. So there is some intangible value in conducting research in general. So I think that I have made millions of mistakes, and I have hopefully learned a little bit from some of them. You know, you find out why this got wrong, and therefore, hopefully you don't repeat that in the next experiment or the next study, or you have some starting point that you can say that I can do that better next time. However, there's a big risk in believing some of these results that are refuted, and many of them even stay entrenched in the literature. So we have done empirical studies, and other colleagues have done the same, where we have looked at papers that have been thoroughly refuted. There's no doubt that that claim or the hypothesis, whatever interest it might have had, it's not really correct. Take, for example, beta-carotene as chemoprevention for cancer. Nature paper, 1980, by great authors, and several observational studies suggesting that beta-carotene may be a very effective chemopreventing agent, that you can get rid of cancer if you give people beta-carotene. 6 big randomized trials showing absolutely no benefit, if anything, harm at high doses. Clear cut that that's not the case. Nevertheless, the Nature paper continues to be cited, even heavily cited, even more heavily cited than the average paper published in Nature in that same year. And if you look at what the citing papers say, they say that beta-carotene is an effective chemopreventing agent, and therefore in this paper we describe the results of some experiment testing a beta-carotene-related compound to cure or preemptively treat tumors in the tails of rats. And it sounds as if this refutation that took so much work and hundreds of millions of dollars, and we clearly know that that's not the case now, is completely ignored. Then there's other cases where we have very clear refutations, and people continue to use the old research as citing evidence to continue some line of research that is congruent with that. And they don't ignore the refuting evidence. They mention the refuting evidence, but they say, we don't believe it. It was wrong. And easily one can think of ways to raise arguments that that trial wasted $1 billion and they got it wrong. They got, well, but you know, that trial, lots of scientists thought about that. Everybody could comment on its design. That was the best shot that we could take. It showed no result. Do you want another one? Sometimes we get another one, you still get the same result. And some of these old hypotheses that have been, or findings that have been refuted, still remain in the literature. So it's not completely harmless to have wrong results circulating out there in large amounts. I think that there's some risk that it's like an epidemic. You can get people easily infected, and it's very difficult to get rid of the infection then at the second stage. So then the second question was about the
hypothesis.
Yeah. So clearly there is an incremental progress. And I think that this is what is wonderful about science, that we have a basis of knowledge that keeps improving. It doesn't necessarily improve in a linear fashion. It could improve in incremental steps. It could include with disruptive big steps. I showed you these data that we have both evolutionary and revolutionary components. Sometimes it may regress. So we have big periods in history where scientific knowledge regressed, like the Middle Ages, for example. You had the Library of Alexandria with 1 million volumes burning to the ground on multiple occasions until practically nothing was left. So I think that even though we don't have situations of Libraries of Alexandria burning in 2015, it's an open question for each scientific field what the trajectory is going to be, whether it's going to be incremental progress mostly stagnation, sometimes even regression into hypotheses that don't pay off, but people just keep doing more and more of that, or really disruptive innovation with big advances. So I cannot give you an answer that fits to all, but I think we need to keep it in mind that the fact that on average science makes progress does not translate that in the short term, in single fields, in isolated communities, we would necessarily see progress through that process.
Question?
Hi, yes. You mentioned that randomized clinical trials have to be registered in order to be published in a top journal. I had two questions. First, could you please recap the mechanism whereby this registration takes place? And second, what do you think about fields that are not at the core of medicine, biology, that may, for example, be more involve treatments that are behavior-based, such as psychology or speech-language pathology? Is registration required, or should it be encouraged to grow, enable these fields to grow in standing and contribute most productively to the greater body of research?
So currently for randomized trials of drug interventions or biologics, they need to be registered before they start. So one has to go to clinicaltrials.gov, for example, and there's other registries as well, and enter some key information about what that trial is about, what type of population would be recruited, what are the compared arms, what are the primary outcomes, where is it done, who is responsible. So some basic information. This doesn't amount to an entire protocol. So even that information leaves substantial opportunities for free lunch of analysis being creative or exploratory after the fact. But at least we know that that trial exists, and we know that 20 other trials exist, and if we see one of them published, then we have to say, wait a second, there's another 19 or 20, and maybe we should wait before we interpret the final conclusion, because maybe the fact that these have not been published, maybe they're still negative, and it's a very different result that they have found. So I think it's a step in the right direction, and clinicaltrials.gov also has the ability to host results of trials once they're completed, and actually there's a regulatory requirement that results would be posted within a reasonable period of time after their completion, although most trials still don't post their results, but at least the regulatory framework and even penalties for that are in place, but the penalties have not been implemented yet. For non-drug or non-biologic interventions, including behavioral interventions, the situation is more tricky because there's not the same level of regulatory pressure. And recently, we published an analysis in the BMJ where we looked at or tried to estimate how many of these trials are actually registered and how many of those are published. So the conclusion is that the large majority are still not registered. So trials of behavioral interventions, lifestyle, diet, surgery, even devices, or other things that are not drugs, the majority are not registered. Probably the majority are not published. And I think that this creates kind of an uneven ground. I think we need to pay a little bit more attention on that. Maybe some of these trials have fewer potential conflicts of interest, and maybe they have less frequent sponsoring by the manufacturer, someone who wants to make money out of that research. But still, the fact that there's less transparency is something worrisome, and I think that it's something that we could fix.
Any other questions?
Hi, I was thinking there are more epidemiologists and observational scientists than there is support for funding for projects that are prospective and very large. So if there were no more underpowered studies going forward in an ideal world, what would the current workforce do of epidemiologists and observational scientists? What would they do And how would you re-engage this workforce to make the world a better place?
Goodness. So now you want me to make 14 million enemies or so by saying that we only need 1 million science instead of 15 million science. Well, I mean, this is one answer, but I'm not going to say that. I don't have a good answer to that. And I think that we clearly need to know who we are and what are we doing. I think that much of the time we assume programs or initiatives to launch training or support one field or another without a very clear anticipation. So what are these people going to do eventually? I think that having lots of people trained in science and working in science is fabulous. So I don't think that, for example, of course I would argue that we need more funding for science in general. I mean, the fact that there's inefficiencies doesn't mean that we should cut back funding on science or we should not educate even the general population about science. Probably we need more of that, not probably. We definitely need more of that, not less of that. But how exactly to structure the scientific workforce is a completely open question. What we know at the moment is that out of these 15 million people that I showed you, which just includes 15 years, so if we take a broader time period, probably it's 20 or 25 million. About 1% of them account for 40% of the published papers and about 85% of the highly cited papers. And 50% of the 15 million only publish a single paper and then they disappear. So that includes very different types of people who have a very transient engagement in science. It could be students at any level, undergraduate included, or medical students or fellows or residents who just take a shot and they publish a paper or they co-author a paper, and that's not what they do after that. There's lots of programs that make it a prerequisite that, for example, clinical residents or fellows, they need to publish papers in order to graduate, which to me is a very dangerous thing to say. You have someone who's committed to doing clinical work, and they have to work 80 hours a week and very hard physically and demanding, and you impose that you definitely need to publish a paper. I would say we should impose that they should definitely know how to read the literature, how to understand scientific concepts, how to judge evidence-based issues, but why should they necessarily publish papers? And there's this type of misconceptions that I think some of them are easier to fix, others probably are more complex on what type of scientific community eventually we want. But I don't think that just eliminating people from science is the solution. It's clearly.
Thank you.
Question over here.
Yes. So I would say that public confidence in science is at a crisis point, and this just contributes to that, of course. But your list of solutions is a little bit overwhelming. And so if you could prioritize 3 things that you think universities could do to contribute to change, maybe you're change 1 and not change 2, I would appreciate hearing your thoughts.
So now you're asking me to become an expert with little evidence. So one, I think that universities could perform studies, ideally experimental studies, to test different policies on how they work, instead of just saying, oh, we will change this or that, but with very limited evidence. 2, I think that universities are at the centerpiece of the reward and incentive system. So they don't control funding that much, although they do have some funding in some cases, and some rich universities have even substantial funding. But they control promotion and selection of faculty and advancement of faculty careers. So I think that they can really concentrate on aligning that PQRST to how they judge and eventually promote and advance the careers of their faculty. This is something that can be a hot debate on every single case, of course, and each scientist is different, but there are some key principles. So if one pays attention to quality, to reproducibility, to sharing, citizenship, translational potential for applied sciences, rather than just number of papers, I think that that immediately would go quite a long way towards really guiding scientists on what is most important to do, rather than just generate yet another handful of papers to be promoted. And then the question is how much universities could do in terms of supporting the reproducibility agenda. And depending, I don't think that all institutions are at the same ground regarding this and they have the same capacity, but maybe there are targeted opportunities that may arise to really enhance some of the reproducibility or even perform some of the reproducibility checks for specific types of research that may be of great interest to the university or a central endeavor for that university in particular.
Okay, last chance. Anybody want to raise your hand?
So as a cancer epidemiologist, I really enjoyed your example of beta carotene, okay? And it's suddenly in my mind, then your example that it still proceeds to be accepted as being protective, created a link with something that I just read like about 2 weeks ago, and I don't remember that article. But exactly, but I mean, I have it. The point is that we as humans react differently to evidence. And there are subgroups of individuals, that article said, that once we form a belief, it doesn't matter how much evidence is presented against that belief. We are actually become more entrenched with our belief than once that counter evidence is presented to us. So that is why, I guess, other studies continue to perpetuate the protective effect of beta carotene. Not because it needs to be, but because we are all just humans and we have certain beliefs.
That's definitely a valid point. I cannot counter that. But I think that means that probably we can learn a lot from disciplines that specifically study these behavioral elements. So perhaps some of the best solutions may come from work in psychological science or behavioral economics, especially in thinking on how we should realign the reward system and what people want to do and what they want to hear, and how they're likely to be better committed to doing better quality work. And some of the results of this research may even be paradoxical to what one may think. For example, I've heard of several colleagues proposing that we don't need rewards, we need penalties. That you would need to pay that much if you do something wrong, which is not fraud, but it's a suboptimal research practice, cutting corners or. We have some evidence from some other fields, like daycare, that penalties could have a paradoxic effect. So there are some studies that suggest that when parents had to pay a penalty whenever they were late to pick up their kid from daycare, then the rates of parents who were late went up a lot. And the reason was that somehow parents felt that before the penalty existed, I'm responsible. This is my kid. I feel ashamed if I do that. And now, I'll just pay $20 and that's okay. I'm not to be ashamed. So it's pretty much the same with scientific processes. If we do something that is extraneous to why we do science, And it becomes just another checklist, or maybe, okay, I'll pay this $20 equivalent. It may make things worse, because then people will say, oh yeah, I'm going to cut corners, fine. Yeah, I'll pay $20 or whatever is the equivalent, and I'll publish an analysis that is suboptimal. Who cares? I have paid my dues to publish a suboptimal analysis. We have to be very careful, and I think that this is why I believe that in metrics in our new center, and we tried to see whether we can connect people from very different disciplines who are thinking about these possibilities. And who knows, maybe if you get an astrophysicist and a behavioral economist together, and you have the answer that I cannot give you today.
All right, I think we have one last question.
Hi, yes. You had a slide where you mentioned about industry-funded research and the, I think you said 96% positive results published, and that, you know, being in this alarmingly, you know, skewed sort of population. But I think that that's the exact same percentage that you said the overall PubMed results had for all data, or all papers published that had significant findings, right? So I think, to me, I guess that kind of emphasizes that industry really has the same level of publication bias, and that maybe it's more of a reflection of how hard it is to publish anything that's not positive data, and that really we have no idea of how many studies are going on that are having negative results, and that really the incentives for both you know, industry-funded research and then also academic research seem to be similar. And I'm wondering, is there really a researcher who doesn't have a monetary vested bias or interest in research? And do you feel that the way that we approach this in industry versus academic research are different problems, or is this all just one and the same?
Interesting observation. So the 96% for the entire biomedical enterprise is the entire biomedical enterprise. If you look for clinical trials, the rates are much lower. So on average, it's close to 50%, and Ben Dulbekovich has done lots of work on that for the common type of clinical trial, which is what we call a superiority trial. So the common type of a clinical trial, you try to prove that a drug is better than nothing or an old drug that is the comparator. And about 50% of the time, you get a significant superiority. So the 96.5% for the industry-sponsored trials, these are non-inferiority trials where the successful result is not a significant difference. It's a non-significant difference. But with a confidence interval for the uncertainty that we have that is not very big. So we say that at the worst scenario, this drug is still good enough. It's not far worse compared to the comparators. So it's a very different type of design. And 96.5% of the time, the conclusion is favorable for the industry. It's actually a non-significant result that is favorable. And that's the paradox here. Maybe I was too extreme about industry trials. I don't want to paint a picture that industry is just trying to cheat, and you know, there are all these fraudulent people who have put together their capital and they want to grow it bigger. And clearly not. I mean, we need the industry to create effective treatments and to then distribute them. But the question is whether this can be done with clinical trials where the sponsor is judging their own product. It's like you have a painter who has to show their paintings in a gallery and also award the prize for the best painting. I think that this is a bit incompatible to happen objectively. And I believe that the industry would also benefit by removing the burden of running clinical trials on their products themselves by having public agencies do that and unconflicted people do that, so that they can use much of that same amount of money, which is currently fragmented along lots of trials that they have to perform, more like marketing tools, like, you know, here's more trials on that drug. They can save lots of that money and use it for real discovery and development of new exciting products.
Okay, well, let's thank Professor Ioannidis.